transition into our next speaker, which is Dr. Eric Wang, Associate Professor in the Department of Molecular Genetics and Microbiology at the Center for Neurogenetics. His laboratory uses just about every technique, molecular, cellular, and computational approaches to understand neuromuscular diseases. He has a personal interest in these disease and has affected family members. Eric works with many nonprofit groups, including very recently the FSHD Society, and he used his expertise to help review some grant applications and help the scientific advisory board. So we really appreciate his contribution there as well. And he's going to provide an overview of FSHD research and various therapeutic approaches. So thank you very much for joining us, Eric.
Great, thanks so much, Jamshid, and thank you and June and other organizers for inviting me to share some of our thoughts on, the kinds of FSHD research that are happening here. But really, the presentation is going to be more of an overview of a number of activities more in the basic science side of the field. As was mentioned, I do have a personal connection to a different muscle disease called myotonic dystrophy. But what it's done is definitely led us into learning more about other muscle diseases and starting to do some work in the FSHD space. We find that learning as much as we can about basic mechanisms underlying disease pathogenesis and just how our cells work really can transfer across different fields. And so I'm going to go pretty quickly through this basic genetics background, because I know a lot of the presentations in the morning did a fantastic job introducing FSHD genetics, basic central dogma. I know other speakers later will also touch on this, but I'm hoping to provide a few tidbits that I think will give you a different perspective on how we think about genetics. And so, as you can see here, this just looks like a bunch of text on a page, but you might notice that the only letters that are here are A, T, C, and G, which are the building blocks, the nucleotides that make up our whole genomes. And you may even remember this image from a previous speaker showing that these letters or bases are organized in chromosomes, and we all have 23 pairs of chromosome. One thing I find is fascinating and incredible is that the length of the DNA from a single cell, if you stretched it out, it's about 6 feet long. So all of that fits into a tiny cell, of which you have about 30 trillion. So if you were to take the DNA from all your cells and stretch it out end to end, it really would span about 2 solar systems. And all of that has to be packaged into your body, which is not that large. So how does that happen? Well, so if you think about how DNA is packaged, we think of it as letters, but it's actually in the form of these molecules made up of atoms that end up getting wound together like beads on a string in these proteins called nucleosomes, are made up of proteins called histones. And then that's really packaged together in higher order structures. If you imagine sort of how a telephone cord, it gets wound as you increase it shrinks as you increase the winding. That's a one way to think about how all this material is packaged together. And as you zoom out more and more and more, there are different ways that the DNA material is packaged. Some is packaged more tightly, some is packaged more loosely. And those sorts of configurations all combine to form what we really can see at a zoomed out level as chromosomes. And so chromosomes, though, have certain parts of them that have unique properties. So that part in the middle, the centromere, actually is linked together at certain times in cell division. And then you have these parts that are also unique at the ends called telomeres. And your body has to do, your cells in your body have to do special things in order to keep the ends of chromosomes intact at the telomeric region. There are special pieces of machinery that allow the DNA to not get lost through cell divisions. And one of the important things about these regions is that they commonly are silenced. They're actually closed up. They're a term we use called heterochromatinized. And interestingly, the genes that are associated with FSHD, one of them, DUX4, is found in a telomeric region. And we think about, whether DNA is open or closed, whether it's in heterochromatin or euchromatin. And that actually has really important implications for whether certain gene sequences can be on or off. When DNA is open, when it's in euchromatin, transcription factors can bind and recruit something called RNA polymerase. RNA polymerase actually copies the DNA into RNA, and the RNA gets made into protein. And so that decision about whether DNA is open or closed is very important for many processes, and particularly important in the context of FSHD. So in terms of genetics, my lab studies different genetic variants. We focus on something called repeat expansions, but just to back up a little bit, when we think about genetic variation, there's about 4 to 5 million variants if you were to just pick 2 random people on the earth between any 2 people. So there's quite a bit of variance, but it's still a very small proportion of the 3 billion bases that make up our genomes. Each child typically will have about 70 new variants that their parents do not have. I know that other speakers have also gone into genetics of FSHD. I'm hoping to offer yet another analogy that might help people to understand a little bit, because the genetics are pretty complicated in this disease. One analogy that one of my colleagues, Christopher Pearson, uses is he visualizes the genetic code in the form of the sentence, the fat cat ate the rat. Imagine that's part of the DNA. When that information gets read, you have this image of you have a cat and you have a rat. But if you have a specific mutation or a variant, if that T becomes a P, then you get a very different sentence. You get a very different idea that comes out of that genetic sequence. In the context of FSHD, you've heard a lot about the DUX4 locus and how there's this repeat. So one way to think about the repeat, it's you sort of have this CA that repeats over and over again. And in the context of FSHD or FSHD1, really, that repeat gets shortened, and there's a change that completes the word cat. And so previously there was no cat, now you have a cat, and that's a problem. And the cat really in this context is this DUX4 protein. And I am oversimplifying this. Really, the situation is that a lot of the sequence is normally silenced. It's grayed out, it's not read, but with that repeat shortening, it allows the location, the gene to be unsilenced, active again, and then that additional T gives you the ability of the cellular machinery to create DUX4. And so DUX4 has been more and more extensively studied in different contexts. This is a movie of a recently published crystal structure of DUX4, the DNA binding part of DUX4 in complex with a DNA target. And a couple of things that are important about DUX4. DUX4 is typically not expressed in mature muscle. It's not supposed to be there. And more research is showing that the activation of a lot of genes by DUX4 is in fact toxic. That is likely what is driving a lot of the pathology in the disease. There are still a lot of details to work out in terms of, which particular genes downstream are toxic, which ones aren't, which ones are really drivers, which ones are passengers. One additional feature that's very interesting about DUX4 is that it appears as though it can actually activate its own expression. So there's this positive feedback loop that is both a problem in the disease, but also a potential opportunity for therapeutic intervention. And so in the context of FSHD, in the context of DUX4 and this toxic protein that's getting made, one of the things I'd like to highlight, and one of the areas that my lab really thinks about a lot, is what a muscle cell really looks like. A muscle cell is quite unique in our bodies. Most tissues are made up of cells that are separated. They're really, each cell has a single nucleus, the command center, and they make up a whole tissue. But muscles are unique in that they're actually a compartment filled with many different cells that fused at some point. And new cells continue to become fused. They're satellite cells. They're cells that continue to come in and fuse with the muscle when there's injury or when there's repair that needs to happen. And so a muscle fiber is really made up of a number, hundreds of cells that have fused together. When you visualize, say, a mouse muscle fiber that you take out of a mouse and look at those individual nuclei, you can appreciate it's really a long tube with a lot of nuclei inside. And again, those nuclei contain the DNA the DNA that encodes for all our genes. If you zoom in further, and this is something that my lab focuses a lot on, we're really interested in, the architecture and mechanics of how these different cells come together and fuse and generate RNAs and proteins. And you can appreciate here, there's a lot of intricate organization to the muscle cell itself. There's a lot of contractile apparatus that allows your muscles to actually contract and move. And then there's a lot of other cytoskeletal machinery that allows for transport of material. And we're really interested in looking at RNAs and proteins. And here, this is an example of an image we can obtain with a high resolution microscope, and we have techniques to label and visualize individual RNA molecules. So here we have labeled a particular RNA called Titin. It's one of the most abundant RNAs and proteins in muscle cells, and you can appreciate that they sort of disperse throughout the muscle compartment. And we do experiments like visualize these RNAs and watch them moving around in cells, and we can track them. And what you can see here is that in a myotube that's been differentiated outside of an animal, you can still visualize a lot of these materials moving around. And I'm bringing this up because I think something that's unique about FSHD and about DUX4 biology is that there is this idea that there is a very small subset of nuclei in the muscle that is making the DUX4. It appears as though it's probably less than 1% at any given time. But the DUX4 protein itself actually can travel around the entire fiber, maybe to adjacent nuclei, and potentially activate itself. And this is really what is causing these pathogenic cascades to happen. It's what's causing the toxicity. It's what's causing the muscles to have dysfunction over time in individuals living with FSHD. I want to introduce just briefly another analogy, and the way I'm going to introduce therapeutic strategies is in the context of this analogy where we think about a muscle fiber. I've been doing some yard work, so I was thinking about how to describe this, and really I thought about how a muscle cell kind of looks like sort of a garden with like a lot of different flower beds, and really the DUX4 protein can be thought of as these invasive weeds that are sprouting out at different times, sort of low frequency, but they themselves have seeds that come out that may spread to other flower beds and cause problems. So really the problem is you don't want this weed growth. And so I figured I would go through some of the ongoing therapeutic strategies in the field, both using this analogy and then discussing briefly the specific modalities that are being used to take these approaches. So sort of starting from most upstream, and then I'll go to more downstream. Upstream in terms of the mechanisms of pathogenesis. So we know that the DNA, there's a variant that causes DUX4 to be able to be expressed, right? So there are efforts out there that are probably earlier in sort of the drug development and clinical pipeline to actually try to change the sequence of the DNA so that DUX4 can't be made anymore. That you may have heard a lot about things like CRISPR, these gene editing approaches like CRISPR. This would be one approach that is being taken still more in the basic research side, I would say. But the way to think about it is kind of like changing the DNA that encodes for the weeds, so they can't even grow. Another approach that's being taken, and you're going to hear more about this from Dr. Sverdrup and who's done phenomenal work in this area, is really how do you prevent, given that genetic variant, how do you still prevent the DUX4 RNA from being made? So this is kind of like preventing weed growth. You're putting on some weed killers. And there's a lot of approaches to try to do this. Small molecules is one of them. There might be approaches using non-editing based CRISPR approaches, other sort of molecular manipulations that allow you to do this. And then another approach is, okay, let's say you can't prevent the weeds from growing at all. You still can try to pull them before they become a big problem, right? So destroying the DUX4 RNA that encodes for the protein. This is an approach that's also being pursued by a number of different parties. You're going to hear about this from Kelly, Teacher Trapani, in a couple talks, where specific modalities that can target RNA and destroy RNA of a specific sequence are being developed. Another approach that's also being taken is to find inhibitors of the DUX4 protein. So you sort of let the DUX4, you're letting the DUX4 RNA get made, and the RNA is also making the DUX4 protein, is getting made into protein, but you're finding ways to prevent the DUX4 protein from actually doing bad things. This is sort of like adding other defective seeds to compete with the real seeds, or dealing with the weeds in some way, preventing their seeds from spreading, right? There are approaches like protein dominant negatives and other small molecules that have been developed to try to prevent DUX4 protein from acting in a toxic manner. And then finally, in reality, a lot of treatments for many diseases actually are quite effective when they address downstream consequences of this whole pathogenic cascade. So this is kind of equivalent to covering the weeds with other plants or dealing with the consequences of the weeds. And these, while they're not as upstream, in fact, a lot of modern day medicine really relies on these sorts of approaches, and there's a lot of approaches out there that are being tested for FSHD, small molecules, antioxidants, a whole class of different approaches. This can also include sort of non-drug or medicine based approaches. As we heard from other speakers this morning, other strategies, occupational, therapy, and other, healthy eating and other interventions that actually can have very profound benefits. And so not to discount those from the context of overall therapeutic strategies. So I'll just leave you with sort of the message that all of these therapeutic strategies require pretty efficient delivery of something that's safe and effective. But something that's unique about FSHD, and I think that's getting more attention from scientists, is that it has this interesting property that it's actually not expressed very frequently, it's not very widespread in muscle. It's very, very infrequent. So that can be a positive, or it can be a negative, depending on the specific modalities, right? So there's not a lot to get rid of, but your therapy does have to go find the target and has to go eliminate it. There's tremendous activity, as you heard from Donovan, about development of muscle biomarkers, both from imaging perspective as well as molecular standpoint, to assess disease progression and therapeutic outcomes. And of course, the involvement of patients and families in that process is absolutely instrumental and critical, and very, very much appreciated to really be able to help accelerate all of the activity in the space. And so, finally, it's a really exciting time in research. There's a lot of approaches that are being actively pursued. There's a lot of successes in other fields that are actually carrying over. There's lessons that are being learned across different diseases. And so it is a very exciting time. But of course, we're all waiting for the day when we do have truly safe and effective treatments. And it's a journey that requires, all these different parties to work together. And so I will be happy to take questions later in the Q&A session, but I'll hand it over to our next speaker.